my god! I am here with Telly! Telly from Telly's Marine Tales.
Yes,
Telly's Marine Tales. Alright, so I have a good story about that. Is that before I started my channel, I wanted to have a name, a channel name that had marine in it, because my name is Marine. Right? And I came up with Marine's Tales. And I was like, no one's gonna be like Marine's Tales. And I think that might be how I found your channel.
Oh,
It was like, Marine's Tales. Damn it, who is this girl? Telly's Marine's Tales.
I got there first.
Yeah, you did. So, it's so nice to be with you, Telly.
Yeah, thank you. It's great to be here.
Yeah. and so Telly, aka Chantel, is here in Manchester with me from South Africa.
All the way in South Africa.
All the way from South Africa. So you're visiting people in the UK, your family?
Yeah. So in my last video, I chatted a little bit about this, and I just handed in my PhD thesis, and my stepsister was getting married in
Congratulations!
Thank you.
Doctorate, Telly?
Not yet.
Okay,
It's already submitted. But yeah, my stepsister was getting married. My dad lives in the UK. He moved here last year, so I decided to come and spend some time in the cold.
Yeah.
It's very cold here.
In the cold, it's not that cold.
It's cold.
It's not that cold.
Compared to South Africa, it's freezing.
So you are going to Scotland in January.
that will be even colder.
Yeah, and so it's so funny because I saw that Telly, you made this video and you said, I'm going to the UK for 3 months, and I thought, oh my god, I live in the UK, we need to make a video together. And so I wrote to you, and then you said, yeah, sure.
I'll come to Manchester.
Can I sleep over at your place? And so here we are, and now we're best friends. That's so nice.
Yeah.
So, well, for whoever doesn't know who you are, I have doubt. I think everyone knows who you are. But who are you? What do you do?
So I am a marine biologist, as you might have guessed.
Yes, because you stole my name.
Yeah.
I have guessed.
I come from South Africa. That's where I grew up. That's where I was born. That's where I did my studies. And as I mentioned, I've just finished my PhD now. So I sort of went straight from school. I knew I wanted to be a marine biologist. I went to university. I studied marine biology and ecology and evolution. And yeah, I did my postgrad work in stingray ecology, which I really loved. I love stingrays. They're really cool animals.
I've seen lots of videos and pictures, and it's pretty cool.
Yeah, and the best part about them is that they're so understudied. Really, people don't know much about them. So any sort of research you do on them is novel, which is really exciting. As a scientist, I mean, that's the whole point of a scientist, is you want to discover novel information. And with stingrays, it's easy because nobody knows anything anyway.
And you're in a field where nothing has ever been done. Yeah. That's really cool.
Yeah. So they're really exciting to work on, and I'm really happy that I managed to get my postgrad work in stingray ecology.
Yeah. So how does it feel to be done your PhD then?
It's a really big relief. It was quite a short PhD as far as they go. It was only 3 years, which is sort of the minimum that they can be. Sometimes they can go on much longer than that. Yeah, like 15 years. Yeah. But the last 6 months have been incredibly stressful, trying to get it all finished and write it all up. And so it's just a huge relief to have it handed in and submitted and just update.
As we were discussing, as you mentioned in a lot of your videos as well, you work from home.
Yes. Yeah.
So you're alone all the time.
Yeah. I'm a correspondence-based student, so I work from home. I don't go into a lab, which can sometimes be... I mean, a PhD in itself is already quite a lonely experience because you're very specialized and you're the only one doing what you're doing. So already it's a lonely journey. And then to add on top of that, the fact that you're working from home, it was very isolating.
Yeah, it's crazy.
Yeah. So my supervisor was really flexible, and he said to me, I'm very happy for you to work at home as long as your work output stays high. So as
Yeah.
long as he could see I was doing the work, that was
Yeah.
fine. He said if I started falling behind or if he wasn't satisfied with what I was doing, then I would have to
Move there.
move there.
Yeah.
So I just made sure that I kept my work quality and output high, and
Yeah.
he was very happy for
Yeah.
me to do it. And also because I didn't have a lot of lab work, you know, there wasn't a reason for me to be there. My
Yeah.
field work was overseas in the Seychelles, so I
Yeah.
would go on field trips there. Somebody has to do it.
I don't.
you know?
Somebody has to do it. I don't even understand these tropical marine biologist people. But anyway, carry on. The Seychelles.
Yeah.
It's just...
So I got to do my field work in the Seychelles.
No, you didn't.
Yeah. I was very lucky. I was very, very lucky.
Yeah.
So I did my field work there, and I had some samples that I needed to analyze in the lab, but it wasn't very complicated
But
analyses.
you analyze them at home.
At home, so
Which is...
so we essentially pumped the stomachs of the stingrays to get their stomach contents to try and figure out what they were eating.
Lovely.
Yeah,
That sounds lovely.
delicious, really delicious. And I would take my samples home with me, and I would sort of sort through them at home and to see what they were eating. And I didn't need a lot of specialist equipment for that. I needed a small dissecting microscope and, you know, some...
So you had your own lab at home. So basically your home is turned into a kind of a university office slash lab, right? So you're doing all your data analysis, everything, your work, your write-up at home.
Yeah.
And you've got your lab equipment, your dissecting tools, your
everything.
Where is your microscope? Is it in the kitchen?
It's on the dining room table.
Oh,
usually.
that's gross.
I do give it a good disinfecting before we eat dinner.
That is absolutely disgusting.
But now the problem is I have no place to store these samples. So they're stored in 70% ethanol, which is incredibly strong smelling, and the stomach contents themselves are incredibly strong smelling. So I have them up in like the spare room cupboard, and every time you open the spare room cupboard, you get this whiff.
Wait a minute. So you told me the other day you should come to South Africa. We have a spare room. Wonderful. So okay, now I understand.
You're a scientist. You did stomach contents too.
I did, which is exactly the reason why I do not want to sleep in the same room as where the stomach content are being stored. So just so it's clear, we're talking about fish stomach content. So here's stingray stomach content, which is basically stingray vomit.
Yeah.
And this
is what I have in my spare room cupboard.
That's what you have in your spare bedroom where the guests go, which is great, and it smells when you open the door. Wonderful.
Nobody goes into that cupboard.
Yeah, and that's what you analyzed on your kitchen table. Sounds great. So half digested material.
Yeah.
And how did you? I mean, now that we're into that, how did you? Was it difficult to do? Because I had a lot of difficulty identifying the organisms because they're so small, and they're usually invertebrates, and they're half digested, and they could be hundreds of different things. And so I had so many taxonomic books because I wasn't doing genetic studies at the time, so I had to do it visually. It had to be taxonomy, and taxonomy on half digested invertebrates is
impossible.
So hard.
Yeah. So luckily for me, with the stingrays, they eat sort of much bigger organisms. So you're looking at like really long worms and crabs and stuff that you can see with your naked eye. You don't actually need the microscope, which makes it much easier. And I think also the technique that we were using, because stingrays are continuous foragers, so that means they just sort of forage all the time.
So they just eat all the time.
Yeah. So there's always, at least where I was working, there was always fresh stomach contents within their system. So, and also the method that we employed, pumping out their stomachs, you always got the most fresh stomach contents that came out.
Lovely.
Yeah.
I love this conversation. Biologists.
This is what you talk about as a scientist,
this is
as a biologist.
And that's what we talked about last night in our dinner conversation.
So they're a bit easier to identify, and stingrays often don't crush their food, they sort of just swallow it whole. So
like a lot of fish.
So some of the worms, they were sort of, in half or whatever, but you could still get either the head end or the tail end of the worms, and that makes identification a bit easier.
But could you identify it to species level?
So it was very rare where I could get to species level, so I sort of went to family level. And I did have a contact at my university in Cape Town. His name is George Branch. He's a very senior scientist on invertebrates fauna, and I would send him a couple of pictures and just ask for his advice and his input, and he was always willing to help, which was a huge help. Yeah,
especially when you're doing that in your kitchen. And did you ever, by any chance, surprise your partner by giving him a bunch of worms for dinner?
No, I never took it that far.
Sneaking a little polychaete in there. Bon appetit!
So actually the funniest thing that I managed to use these samples for, because once you've analyzed them, you know, they sort of lose their value. I mean, you still have to keep them just in case, but there's not really much else you can do with them. So, you know, I had stored them up in my cupboard, and in my first couple of years of my PhD, I was a part-time au pair.
Yeah.
And so the one day, it was like in the school holidays with the children that I was looking after, and they were quite old. They were like 7.
I don't know where this is going, but I'm scared.
So they were like 7 and 9 years old, and so I had like a scientist stay with them. So I brought over my dissecting microscope and my samples, and like also my taxonomic books that I had, and we like went through the stomach contents. I made them go through the stomach contents, and we identified the stuff, and it was actually really fun. So that was sort of just working with them and showing them what a scientist does sometimes, you know.
Yeah, in their kitchen.
Yeah,
that's great. Why were you looking at the stomach content of these stingrays? Mm-hmm. I'm sure they didn't enjoy
no, they didn't enjoy the process.
But it's better than killing them.
Yeah.
Right?
So specifically for our project, we were looking at non-lethal techniques. We didn't want to kill any of our animals because they are all listed as threatened by the IUCN. Stingrays in general are really, really vulnerable, and there's a lot of population declines, and the species specifically that I was working on are in trouble. So we made sure we didn't use any non-lethal, we used all non-lethal techniques. And the reason why we were interested in their diet is, as I mentioned before, there's really not much known about these animals. So being able to just simply know what they eat, you know, it's just such a basic question, and it's a question we don't have an answer to. And if you want to start managing and protecting these animals, you need to have an idea of how they live and specifically what they eat, so that, you know, if you're protecting an area where maybe they occur but not where they don't eat, then you're not really helping protect the animals, because you need to protect an area where you know that that's where their food occurs.
Yeah. So for example, you mentioned that there are animals that eat all the time.
Yeah.
So you would expect that wherever they occur, they would be eating.
Yeah. So it's really difficult to know because the literature is so limited. So some species in some places, they have patterns of activity over the 24-hour cycle. So some are more active at night, some are more active during the day. So there is some speculation that they will eat during the day or during the night. But in the times that they are feeding, they are sort of constantly foraging. You know, they're almost like grazers basically, as opposed to a hunter who spots a prey and goes for it, and then can go, you know, for hours or days without any food. Whereas, you know, stingrays, they just constantly graze as they go along, but they do have periods of time where they rest, and they also have to avoid predators, because they are sort of in the middle of the food chain, so they themselves get preyed upon by sharks.
Sharks eat stingrays.
Yeah, so big.
But how big are what size are we talking?
So it's specifically the juveniles. They can be really small. Like, you know, 30 centimeters wide. They're really little and they're really cute.
Oh, that's cute.
And some species also, as adults, they don't grow very big. Some some do get very big, like more than a meter wide. But specifically the juveniles, which is what I was working mostly on. They're very easy prey for sharks.
Are the adults prey for sharks at all? Or?
Not the really big ones.
Not the big ones, okay. Because they also have a sting and they will like...
Yes, yeah. So oftentimes you'll hear of tales of people bringing up a shark and they have sort of a stingray barb in the side of its mouth.
Really?
So they're really good at protecting themselves.
Does the barb actually come off when they...
Yeah, it's like a bee sting, so the barb will come off. Yeah.
But it doesn't kill them?
It doesn't kill them, no. No.
Does it grow back?
Nope. So once they've used it, it's gone. Sometimes they have 2 barbs, so they can sting you twice, but once it's gone, it's gone.
So does it systematically come off when they sting? Do you know if, like, can they sting you and it's not gonna come off?
I don't actually know. That's a good question.
Because I wonder if the stinging is kind of like a one in a lifetime opportunity. So you really need to be, holy smokes, this is the end of my life, I'm gonna sting this guy.
But that's exactly why they're actually not all that dangerous, because they know that it's sort of like a one-shot wonder. So they will first try and swim away. So first they hide. Their first line of defense is to stay hidden, and that's why they live on the seafloor and they hide themselves in the sand. Their second line of defense is to swim away. As soon as they sense any sort of danger coming to them, they will swim away. And if they really feel like they're trapped and it's their last resort, that is when they will use their sting.
Okay, so as a scientist, even though you're not killing them, when you're catching them, you want to make sure that they're not going to attempt to sting you. Because then they'll lose their barb. And then
you'll be hurt. The stingray will be without its protective mechanism.
And then the stingray will lose its protective mechanism.
So we were very careful about how we caught the stingrays and how we handled them. So the small ones, we sort of used dip nets. So because they're in really shallow waters, like waters that deep. So you could come up to them and you could use a dip net and you could catch them that way. And then we would have we had a set of chainmail gloves. So somebody would then come...
Where do you get chainmail gloves? I saw on your blog that there's this guy wearing a whole chainmail outfit, and I was just thinking, holy smokes, Halloween outfit right there!
So I don't actually know where it came from. It was just part of the equipment at the research station that I was based.
It was stolen in the museum. Like, do these exist?
They exist.
What do people use this for?
I don't actually know. Like I said, I don't really know why they had it at the research station. But they were there, and they were really useful for us as well.
That's amazing.
So with some of the bigger stingrays that we caught, with the person who was at the front of the sting, because if you have your stingray and you have the tail here, oftentimes the tail will come up and sting the person in front of them. So that's actually where the most danger is.
Oh, really?
So it was the person who was standing in front of the ray.
Kind of like a scorpion.
Yeah, exactly. Yeah. So the person who was standing in front of the ray, who was sort of because how we would catch the rays, somebody would sort of hold it in its mouth and then hold it on top so we could flip them over like a pancake. So that person doing that job was in the most danger, and when we worked on the really big animals, that was the person who wore sort of the whole chainmail suit in the front, just in case.
That looks so cool. I mean, it must look so cool.
But before we even got to that stage, we would make sure that somebody was handling the tail end. So that was the person wearing the chainmail gloves. And
someone's grabbing the tail?
Yeah, so somebody would first grab the tip of the tail, because the barb is sort of halfway up the tail. So they grab the tip of the tail, stabilize the tail, and then use their hand and wrap it around the barb and the tail.
Oh, so that sounds so scary.
I mean, that's not really the dangerous part, because once you've got it, it's not going anywhere and it can't sting you. So somebody then would hold the barb against the tail and would hold the tail steady so that it couldn't go anywhere.
Is it hard? Like,
they thrash around strong? A lot, so
I guess the bigger ones are stronger, obviously.
So strong, and they're so heavy as well. They're so difficult to lift. But really, their muscle is in their body. They don't really have much muscle in their tail. So the name of the family is actually called whiptail stingrays, because their tails really move around like a whip. So they're very flexible, but they don't have a lot of muscle. So there's
like something with a whip, and there's a dude in a chainmail, and what are we talking about again?
When you put it like that, it sounds...
Oh, and there's vomit involved. Like, I'm just... This sounds like so much fun.
It was a lot of fun. It was really, really awesome field work.
So why do you flip the rays upside down?
So when you flip them upside down, they go into something called tonic immobility, which is what sharks do as well. So when you flip them upside down, there's some weird thing that happens in their brain, and they sort of become immobile, and they become really chilled out, and they don't move a lot.
I think I've seen that in documentaries. People just and they're swimming with the shark, and they just turn them around, and then they pet them, and they say, "Look at how much the shark loves being petted," and really, it's just paralyzed.
So it works better on the sharks. The sharks, I found, enter tonic immobility a lot quicker and a lot better. So the stingrays will still sort of move around a little bit. They kind of like wave their wings a little bit.
Do you call that wings?
Yeah, I mean, that's not the official term.
What's
but
the official term?
Pectoral fins.
Oh, okay. So they are pectoral fins.
It's just like a modification of a fish's pectoral fins.
That's a pretty cool modification.
Yeah. So they will sort of move them a little bit, but they're not like thrashing around.
Okay.
So they're much easier to work with. And then also when you have to pump their stomachs, their mouth is on the underside, so you need to be able to access them from underneath.
Yeah.
And also that's where we implanted, because we also tracked the animals, so we surgically implanted little trackers, so that would also be on the underside. So basically everything we did was underneath the stingray.
Yeah. So have you ever been stung by a stingray?
No, we had a very clean record.
Wow.
I mean, we caught, I can't remember the exact number, but it was over like 250 stingrays by the end, and we had quite a big team working there, because obviously it's quite difficult. So we would have 4 people in the field at a time working, 4 to 5 people, and not a single person ever got stung. So
wow,
we had a really good track record.
That's really impressive.
As long as you respect the animal and you understand how they work and you are careful and you, as I said, you just respect the animal and give it its space and be as careful as you can with them, there's very limited chance of injury.
Yeah, but you'd expect that, I mean, given the way they're whipping their tail and just flipping it over their body and all that, you'd expect that there might be an accident here and there. But
no, we
that's
were
really
very,
impressive. So how did you catch those stingrays then?
Yeah, so the small ones were with the dip net, so we would sort of just take the dip net.
It's what is
it's like, I'll add some photos so you guys can see, but it's basically like a net that big, and it's got a handle, and then you literally just like scoop up the stingray. The bigger ones we caught with a hook and line, we foul hooked them, which basically means you spot a stingray, okay, that's the guy I want. There's no bait or anything involved. And you sort of cast for that stingray. And my supervisor did, both my supervisors, they are incredible fishermen, and their aim was spectacular. So,
oh wow.
Yeah, I mean, I could never have done that, but they spotted the stingray and they cast, and they, probably 80% of the time they managed to hook the ray.
Wow.
Yeah.
Does that injure the ray a little bit?
So there is a little bit of injury involved. We try and hook them not too close to the edge of their fin, because then the hook can pull out.
Oh, so you're actually the hook is just grabbing onto them on their body anywhere.
Yes, on their body.
So it's like unlike fishing where they just grab it with their mouth.
Yeah, because we weren't using bait. It wouldn't have hurt the mouth or anything. We were sort of just working on the edges of their fin.
So you throw the hook and then
and then you
pull it back and
we try to make sure. I mean, I don't think we ever had any awful injuries, because we avoided the stomach area and the head area, and it was always trying to get it on the edge of the fin where it minimizes damage. And then you pull it in, and I've seen a video where there's this guy running after a stingray as well. Yeah, so the most fun part about this field work was catching the Porcupine Ray. So this is the only species of stingray that doesn't have
a
sting on
Poor
its
guy.
tail. So, and he is the same color of the sand, so he relies very much on camouflage. So you can really sneak up behind them. You can sneak up really close to them, and what you do is you lunge and you try and grab hold of the tail. Dive after the stingray, and grab hold of the tail and try and put your hand on the back so that you hold them flat against the water. And then we didn't have the issue of injuring them with the hook or anything like that. So for them,
so it's better to try and grab them like that.
Really. So it obviously gives them a big fright. But
they're probably thinking, man, why don't I have a hook? I mean, that's
but and also, they're called porcupine rays for a reason. Their whole back is covered in like these really
So they do have something.
They do have their protection. So you have to use really thick gloves to make sure that the thorns wouldn't sort of injure your hand. So it's impossible to catch them with nets as well, because if you catch them with a net, they just get all tangled up in it and they destroy the net, and it's impossible. So I mean, I've had some of other scientists look at this method and say, oh, this is, you're hurting the stingray or whatever, but we found that personally for us in the area that we were working, it was the least harmful way to catch that species of stingray.
I mean, which other way would you use?
So that's the thing. I mean, I don't know. You could try the hook and line, but then again, you're
hooking them. So.
And he suggested nets, and I was like, well, if you've ever really worked with a porcupine ray, you know that nets wouldn't work.
Yeah.
So we just decided that was the best way for us. And it was really fun. It was really fun.
Does seem like
and they're so clever as well, because if you dive the first time and you miss them and they escape, they will swim around and they will flap their wings and they create this huge sand plume and they disappear into the sand plume and they'll hide out for a couple of seconds and then they'll dart off in the other direction. And then you gotta chase them.
That they're there.
And they try and dart off, and then if you spot them you gotta chase them again, and then they'll make another sand plume and hide out and then dart off in the different direction.
So you have to be a whole team.
Yeah, that's exactly why we needed a whole team. Was
because the guy who's like, oh, there's a sand plume here, and he just dives there, and meanwhile someone else is like, actually
there
it's
already.
over there.
And I remember the one time, so for that species there were only really juveniles in the area that we were working, so it was sort of 60 centimeter wide animals. But the one time we managed to spot an adult, and this thing was massive. It was more than 1 meter wide. And also, they're a little bit different to other stingrays because they get really thick as well. So even if
Oh, really?
Yeah. So most
you imagine a stingray to be a little bit flat.
Exactly. So most species, even if they get bigger, they still stay quite flat. But this species, I don't know why, it gets really, really thick.
They've got giant stomachs full of stuff.
Exactly.
Lots of fun.
So I was with, there was a volunteer who was with me. His name was Rhett, and he's much bigger and stronger than I am. So I said to him, you got to go get that adult for me.
That giant there.
And the water we were in at that stage was a little bit deeper. So it was probably sort of halfway up your leg. So it was quite deep. So he goes and he stalks the stingray and he pounces on it, and this thing gets such a fright and it takes off, and he's holding onto the tail and he's getting like pulled through the water.
No way.
It was the funniest thing I have ever seen. But kudos to him, he managed to grab hold of the tail, and eventually the stingray came to a stop, and we got it.
Wow.
Yeah. And it was the only adult we ever managed to catch.
Oh, wow.
Yeah, so
well, he's pretty good.
He was fantastic.
If ever you need a good fisherman.
Rhett Bennett.
Wow.
Yeah.
That's really impressive.
Yeah.
Did you have footage of that?
No, so at that stage, it was right at the end of the day, and we were exhausted, and we were actually heading back to camp. And we didn't have any of our camera equipment or anything, and we didn't expect to see any rays at that time of day.
Or to be able to catch any in that fashion.
And it all just happened so quickly, you know. I was like, there's an adult, I want it, go get it. And so unfortunately there was no footage of that one, but it's forever imprinted in my brain.
There's a good story. Wow, that's wonderful. Well, that's much better than killing them. Have people killed them in the past? Because I know that a lot of researchers do that. I mean, this is what I did with the fish I was studying, because they're not endangered per se, and I mean, fish is one of these things that people don't seem to care about that much. So the ethics on fish are fairly low. The ethical standards, at least. And just like fishermen that are going out there and just throwing their net and fishing, the researchers do the same thing, and then they bring up the fish, and the method is so invasive that they die, and then this is how you study them, basically, which is not the nicest thing. But there's no other way.
There isn't, it's a tough rope to walk. There is an argument for, lethal sampling, because sometimes the information you need, you can only acquire from lethal sampling. And like you say, if you're not working on species that are threatened or anything, really, you're actually doing more good, because you are getting something out of that species.
You are doing some research.
You're contributing to the knowledge. It's more difficult when you're working with elasmobranchs, which are the sharks and the rays, because many of them are threatened. So there's a big drive for non-lethal sampling. Still, sometimes I read some papers about a guy who works in Australia, and he strongly advocated for lethal sampling for rays.
Interesting.
So
he actually advocates for that.
It was part of his PhD thesis, actually, and he had a whole chapter on sort of lethal sampling and sometimes why it is needed.
What are his arguments?
So, I mean, if you're trying to look for any sort of biological estimates or life history estimates, for example, you want to know the age of the stingray. We don't know the age of a stingray unless you kill them. Or
can you tell the age of a stingray? Because I know that's very hard on cartilaginous fish.
It is very difficult. So I mean, even that, there's still a bit of uncertainty surrounding it, but it is the best and actually the only way There's no other way you can tell the age of the stingray. Yeah. And if you want to look at the whole reproductive cycle and all that, unless now there's sort of ultrasound, machinery and stuff, but
it's expensive and not everyone has the technology.
Exactly. So really, sometimes lethal sampling is required. But as a scientist, you always have to go through an ethics committee. I think even you working on fish, there is an ethics committee that you have to go through. So it's not like you can just go out and kill all the fish. You have to have a very good reason for it. You have to make sure you have enough of your sample, but also that you don't take more than you need. You take the absolute minimum.
Often it's going to be a bunch of projects are working with the same sample. Yeah. So there's going to be a trawl happening, and then there's going to be a whole bunch of different people that sample from this one trawl. Yeah. And sometimes you're going to share the fish, or you're going to share information about a given fish. Like I would take a whole bunch of different measurements. I'd take the stomach, for example, and then someone else would do stable isotopes on the same fish. So then they would take pieces of muscle, and you end up where there's not much left of the fish because everyone took something. Which is great. Which is ideal. Yeah, that's what.
And another alternative to sort of lethal sampling is taking specimens from fisheries.
Yeah, which is what I do now. Yeah.
So a lot of times stingrays get caught as bycatch in the nets. So a lot of that information comes from stingrays that were caught in the nets that are already dead, and if there are scientists involved, they can take the animal and do what they need to do with it. So that's actually really mostly where the information comes from for elasmobranch rank research. Is there's really
from bycatch.
There's very limited actual lethal sampling. It's mostly through fisheries or through non-lethal techniques.
And it seems like it would depend on the species as well, right? Because the species you were working on, or at least that life stage, so they're juvenile, and they occur in these very shallow areas, so it's easy to catch them.
Yes.
But then once they're adults and they just go out in the deep, it gets a lot harder to catch them,
it becomes
or even
difficult.
borderline impossible.
Yeah, I mean, you still would get sort of the deeper water trawls that would catch them, because they still benthic, they still live.
But that would be bycatch.
But the biggest, as you say, the biggest threat to them is sort of this interactions with fisheries when they are in their juvenile life stage. They're really shallow waters. Any fisherman can go out on his boat and set a net and catch them.
And just catch hundreds of them. Yeah. So how many are there in that area where you were working? So you were working in the Seychelles, which... Again, too hot for me, no thank you. Blue lagoon waters, turquoise, 25 degrees Celsius water.
Beautiful.
Okay, we have different standards, clearly. She's the tropical marine biologist, I'm the Arctic marine biologist.
I don't do well in the cold. Like, some people do well in the cold and hate the heat. Some people do well in the heat and hate the cold. I'm one of the latter. I don't do the cold.
I don't like the cold and I don't like the heat either. Somewhere in the middle. So what was I saying? So you were working in the Seychelles, and in this lagoon, right? Oh, you don't call that a lagoon. You call that an atoll.
Yeah, so the area
What is an atoll?
So an atoll is basically a sunken volcano. So
it is, okay.
Yeah, so you have this rim around it, which is sort of these shallow water areas, like, between 10 centimeters to maybe up to 2 meters deep, but only really at high tides.
So that's basically the top of the volcano, right?
And then in the middle you must have this
really deep.
So I think it's because it's not really deep. It's only about 10 meters deep. So I think there's a lot of sedimentation involved that closes it off. But there is still that deep crater in the middle that forms the lagoon.
Is an atoll gonna be a volcano that is how do you say that? Extinct. that is not active at all?
I think so.
Yeah. Okay. And so you're working in this fairly shallow area on that rim.
Yeah.
How wide are we talking about? Like, how wide of a rim?
So it depends. It sort of gets narrower and it gets thicker in parts. But I suppose at the thickest, it was probably about 1 kilometer wide.
Okay, so it's fairly big.
It's quite expansive sand flats that you were working on, and it really to try and cover that whole area can be quite difficult.
Yeah.
And some of the places it's really shallow, and some of the places it's a bit deeper. So even if you're trying to maneuver a boat over those flats, it can be very difficult.
Tricky. Is there coral there, or is it just sand?
It's mostly just sand. There's a little bit of coral rubble. There's seagrass beds, so there's quite a lot of seagrass. But then in the lagoon, you have these sort of coral outcrops
inside the lagoon. Yeah. Oh, that's really interesting. So, why are the juvenile stingrays hanging out in those shallow areas? Is it kind of like a nursery for stingrays?
Yeah. So that was one of the hypotheses or questions of my thesis was, do these juveniles stay on the sand flats? Do they move around? Do they go into the deeper lagoon? Do they leave the atoll? I mean, we had no idea.
Yeah.
So that's why we tracked the movements of these animals, and what we found was that really the juveniles specifically spent the vast majority of their time on these shallow sand flats. And my idea is that they live there because they can escape the sharks. So there are a lot of sharks in the area. There are quite big sharks in the lagoon as well.
In the lagoon. Do they hang out in the lagoon the whole time, and can they swim through this shallow area?
Yeah, so because there was another project that was tracking the shark movement. So we had that data, and it was really nice because they would spend quite a lot of time in the lagoon, but then at high tides they would come up onto the flats. And at that time they could leave the atoll, but most of the time they would just come up onto the flats, and that's where they would...
Would you see them? That must be kind of scary.
So we did see them actually. Every now and then, as the tide was coming in, you'd see the fins of these sharks sort of cruising along the flats.
Get out! You're eating my rays. Then so you put this transmitter in their stomach, and then if a shark eats one, if the shark eats it, does it still transmit? Are you still gonna catch
So it will transmit for a couple of days because the ray would enter the digestive system of the shark, and then eventually the shark would poop it out. So it would stop transmitting after a couple of days, but there would be this period. If that happened, it didn't happen with my data, but it has happened a lot before, and you could see this distinct change in the movement pattern of the animal, and you're like,
that's so
what in the world is this animal doing?
So he's like a slow guy who never moves, and then all of a sudden
he's moving everywhere, and then all of a sudden the detections stop, and then he's being...
So why does it stop? It stops as soon as it touches the water, like when it's being pooped out?
So the method that we were using is something called acoustic telemetry, and so you have these transmitters which transmit acoustic signals, and we also have an array of receivers in the area, and it was only when the transmitter came into close proximity to the array, to the receiver, that you would get the detection. So you don't have constant detection. So it was only when an individual swam next to a receiver that you would know that it was there.
Okay.
So if the shark pooped out the transmitter,
it stops moving.
Then it stops moving. It's lying on the bottom of the seafloor, and you don't get
unless something else eats it because it thinks it looks cool and tasty, and then you end up having these weird signals again.
Yeah.
That is so cool that it actually does happen.
Yeah.
So it's funny because you'd think that maybe, it needs to be fairly close to the skin or something like that in order to be received by the receivers. Yeah. But then if the animal gets eaten by a shark and it's in the digestive tract it still transmits.
Acoustic signals are
so actually, they must be quite strong.
Yeah, they're very strong in the water column there. And actually there's a lot of research going on now about how it affects marine mammals, because they also use acoustic signals
for marine mammals.
So how that interacts with marine mammals, because it's at a very low frequency, so we as humans can't hear it, but obviously the marine mammals can hear it. So there's a bit of...
so it actually makes sound.
Yeah, it makes a sound. It transmits a sound through the water column. And that is very easily picked up by the receivers. So the receivers have... It depends on what sort of system you're working in. Shallower areas have a lower range, but if you're in like really nice, clear, deeper waters, they can transmit, like some of the fjords in Norway, their transmission range is like over 1 kilometer, that they can pick up those signals.
That's crazy. Yeah.
Whereas in the area I was working with, it's very shallow. There's a lot of noise from all the animals in the seagrass beds and all the wave action and stuff. So really only about 200 to 300 meters could you get
so it depends on the turbidity of the water and all that.
There's a lot of stuff that affects it.
That's cool. Could you study predator-prey interaction using that? But you're putting these in a bunch of potential prey, and then you somehow you kind of estimate whenever that prey is getting eaten by something else.
Yeah.
Because the movement changes, or
so there was another project.
It sounds like it'd be really difficult to do, but
yeah, it depends on the species that you're working on. So another one of the projects that was happening at the same atoll, the girl was tagging bonefish, which is it's a big sport fish, so a lot of anglers like to try and catch these fish, but they get eaten by sharks a lot. So her project was originally supposed to be trying to track the movements of these animals, but as soon as she released them, as soon as she released them, they got eaten by sharks. So her whole project changed, and it was actually looking at mortality rates
of
these fish after a fisherman has been handling these fish.
Yeah.
So it really just depends on the species you're working on.
Yeah, okay, because then she was handling the fish, so then she's looking at how that affects.
And that fish specifically puts up a really big fight when you are trying to catch it. So it tires itself out. And even though they try to minimize, as scientists, you always try to minimize the handling of the animal as much as possible, because they have this protective mucus layer. And as soon as that's gone, the sharks, they know that fish is there. So, and also they release like stress hormones and stuff into the water.
Okay, and the sharks are really receptive to all of that.
So even though, like my supervisor, he really tried to minimize all that stuff. They had sort of like a floating crate that they would put the fish into so that the hormones couldn't be like released into the water column, and they try to minimize the handling as much as possible, and they try to bring in the fish as quick as possible so it didn't have a really long fight time. They still got absolutely annihilated by the sharks.
Okay, so do you think, so they wouldn't necessarily die when you put them back in the water, they just need their time to get over it, but because the sharks are right there, they're so weak that they just get eaten right away.
Yeah, that's it.
Yeah. So you use these
transmitters.
These transmitters that you put inside the animal. And does that affect them in any way? Like, because they're pretty big, right?
Yeah. So we have different sizes of transmitters. So from really, really small ones to really big ones. And the bigger
really big ones.
No, not really big ones. I think the biggest one is sort of maybe a bit longer than your thumb.
Okay. Which is
It's pretty big.
It would be very fun to have that. Although a lot of people walk around with all sorts of weird surgical equipment.
So you have to be aware of the size of your animal. So the smaller the animal, the smaller the transmitter that you use. The bigger the animal, the bigger ones you can use. And it's beneficial to use the larger ones because they have a longer battery life, so you can get more data out of it. But you have to be mindful of the animal that you're working on and try to minimize the discomfort as much as possible. So we try to make the surgery as quick as possible. My supervisor is he has a lot of experience doing this. He's tagged hundreds of animals, different species, everything. So you just make a quick incision through the sort of the wall of the abdominal cavity, and you just sort of pop the transmitter in, and then he does 2 or 3 sutures to suture them up.
Is it like kind of nested within the muscles, or
no? So they actually have like on their underside they have like a really thin layer of skin, and then it sort of just opens up into their abdominal cavity. So you just make sure you cut through the skin layer. Obviously try not to go too deep, and then it simply just like pops into their abdominal cavity. There's like a lot of space there. So and then you would suture them up, and he had like this really cool gel substance that would sort of seal the wound so that it gave it a chance to heal. But elasmobranchs in general, sharks and rays, have really quick healing times.
Really?
They have really, I don't know what it is, it's some sort of property that they have, but their wounds heal really quickly.
So that's because they just bite each other all the time? Maybe?
Yeah. I mean, we also collected muscle tissue sample for stable isotope analyses, which is just something that we use to get a better idea of what they were eating. And to do that, we sort of just had like a little, it's called a muscle punch. It's
like a puncture.
So it's basically like you puncture the skin and you take about like a centimeter.
They do that for dolphins as well.
And then you sort of just punch the skin out and the muscle out, and you take a little sample. And we had caught individuals about a week or 2 after we had sampled them individually, and that whole thing was already healed up.
Oh wow!
Completely.
Okay.
So they do heal really quickly, and we didn't have any mortality rates with the animals we were working with, and we always monitored them after we performed the surgery, and they always sort of resumed normal behavior. So we were pretty confident
Yeah.
that it didn't have a huge effect
Yeah,
on the
yeah.
animal.
And does it stay in them afterwards forever?
Yeah, it stays in them forever. So there's also an alternative to that where you externally attach the transmitter. So instead of surgically implanting it,
and then it detaches with time.
yeah. But sometimes I prefer the internal attachment because the external attachment in the marine environment, it gets
Creates
full of
drag,
algae and it
yeah.
creates drag, and I think it makes the fish a lot more visible. You have this, if stingrays are flat
this
and they like to be
antenna,
on the bottom
yeah.
floor, and then you have this thing sticking up
Yeah.
that's got a trail of algae behind it.
Yeah, no, that's not good.
So and also then because you have to sort of like attach it to their fin, and so there's always this irritating thing on their fin, and they don't heal. So personally, I prefer the internal attachment because it might be more invasive initially, but it heals really quickly and it's done, and there's no further
it's
impact
just part
on the
of the
animal.
body basically afterwards.
Yeah.
And then how long does it work for?
So
Because it's battery powered.
battery powered. So the bigger
What kind of battery are we talking about? Just like your random AA?
I have no idea.
Okay.
It's literally like this little black box cylinder thing that you put into the animal. So I don't know what it looks like on the inside. But the really big ones, one of the ones that we used on some of our adults, can go for 10 years.
What?
Yeah, it can go really long.
10 years?
So you can track the movements of one animal
No
for 10
way.
years. Yeah.
Wow,
Which
that's
is really
so
cool with
long.
stingrays because they have these long lifespans, and you can,
How long do they live?
So that's also a bit up in the air.
Yeah.
They are really long lived though, so for like 30 years kind of thing. They live for a really long time. So you can, put a tracker in it when they're a juvenile, and you can track their movements as they mature and as they become an adult. It's really exciting stuff.
Provided they stay in the same area, right?
Yeah, so that's also the downside, is that they have to be obviously in an area where there are receivers, where you've put your receivers.
Yeah.
So.
But let's say, for example, that you have that this is a nursery area, and that they stay there when they're juvenile, you tag them, and then they come back systematically to that area to reproduce, for example.
Yeah, then you could pick up that pattern as well.
You don't hear from that ray for a year or two, and then all of a sudden you get the signal again.
Yeah.
That's so cool.
Which is what happened with the one adult porcupine ray that we tagged.
Really?
Yeah, which is super exciting.
Oh, wow.
Yeah.
So it left.
It
So
left.
You tagged it.
Yeah, we tagged
it.
It left. And that's why I think the adults of that species, they don't live in the atoll, because we never saw them there. That was the only one we ever saw. And it left as soon as we tagged it.
Oh, and you're probably thinking, oh, well, it's never going to
We'll probably never get it back. But I think it was after a year and a half, maybe 2 years, we got the signal back that it was back in the atoll.
That's so cool.
Yeah.
That's
so you can't really
cool.
tell what it's doing. I mean, you can postulate that it comes back to reproduce. But you can't say for sure that that's what it's doing.
Yeah.
But it really provides very cool information.
That is very interesting. So what kind of information did you get from tagging the juveniles?
So we were mostly, as I said, looking at whether they sort of stayed in that area, whether they moved, whether they used shallow waters, deeper waters. I also wanted to look at whether the different species were all living in the same area, or whether they lived in different areas, sort of looking at their interactions, and then really determining that it is a nursery is great for conservation efforts, because if you can say that this isolated, compact ecosystem is a stingray nursery, then it makes for a really good reason to turn it into a marine protected area.
Okay.
So that can directly feed back into conservation efforts.
So right now it's not a marine protected area.
So there are plans to make it a marine protected area. It actually was, it was gazetted as a marine protected area in 2014, but there was some sort of legal process that hadn't been adequately followed. So that sort of ruling was retracted. And now, I mean, we're 4 years down the line, and there's still this umming and ahhing and going back and forth of whether they want to make it a marine protected area.
So does it mean that there's fishermen going there and fishing those stingrays?
So not the stingrays. There is a fishing resort on an island that's nearby, and they go there for the bonefish, because it's known as a really good fighting fish. So fishermen love fish it. So they go there for that, and then also just like the trampling on the sand flats and stuff actually leaves quite a big impact. They're walking on the sand flats. And there has been documented cases where people will come and fish out the sharks as well, and sometimes the locals will also sort of fish out the stingrays, but that area is really pristine because it is so isolated, so there's not a lot of fisheries impact. That was one of the great reasons for working on this community, is that it was pristine, it was abundant, there wasn't a lot of fisheries impact. So the information that you got from this community of stingrays, you could then use to look at other communities of stingrays which are more impacted by fisheries and see if their behavior changes at all.
So you kind of extrapolate a little bit and use that as a reference.
So we hope that this information can be used to add evidence that this would be a great area for marine protected area, because then it will protect this community from potential future, it's right next to an island that is privately owned, and if somebody had to buy that island and turn it into a big fishing resort or a big tourist resort, that whole ecosystem could just become degraded very quickly. So to keep it protected.
So were you able to show that it's a nursery area? So
it
is a nursery.
It is a nursery area.
Yay, baby stingrays.
So there is specifically for Elasmobranch nursery areas to define, and you need to meet 3 criteria.
So also sharks.
For sharks as well. And I managed to meet the 3 criteria that you need, so I could...
So what are these 3 criteria?
So the first is that there needs to be more juveniles in a specific area than in surrounding areas.
Okay.
The second is that they need to spend a long time there, so you can't just like sample them there for one day and say that it's nursery. You need to show that they're there for a long period of time, which is what acoustic telemetry is great for. And then you also need to show that they spend...
That they eat a lot.
That they eat a lot,
Really?
But that they occur there for multiple years. So you need to have this annual data to show that different cohorts, different generations use that as a nursery area. Which again is acoustic telemetry is great for, because you can get this really nice long-term data set.
So from the juvenile individuals that you tagged, how long do they stay there for?
So it was very dependent on the species that I was working on. The one species, some individuals, because we tagged them in May, and by the following warm season, which was sort of in January, December.
So is that, are the season reversed as well in the Seychelles? Are we south of the equator?
We are south of, well, actually we're pretty much on the equator. So differences in seasons, it's not really, it's hot all the time.
It's just hot all the time.
But you have a rainy, so you have a rainy, super hot season, and then you have a windy, hot season. It's slightly cooler, but it's still hot, drier. So we tag them in May, which is is the beginning of that cool, windy season. And then by the following warm season, which is your December, January time period, some of them migrated out and we stopped getting detections. And then the following year, in the following warm season, some other individuals left. But it didn't seem to, quite interestingly, it wasn't related to their size. So it was all sorts of sizes. You would think that the larger individuals would be the ones to go first, and then the smaller individuals would be the ones to leave the following year. But it was a bit of a higgledy-piggledy mix. Anybody left. So we weren't quite too sure what then was the process behind that migration from the nursery area. But it was interesting nonetheless. It was actually the first study that really showed evidence of dispersal from the nursery area. So that was really interesting, and to show that it didn't happen at a specific size threshold. It was just I don't know, whenever the ray felt like leaving, he felt like leaving, kind of thing.
When you feel ready for the world, basically.
Go on and take it.
I am now an adult. I shall face all these mean sharks.
So it's quite interesting, and it brings in this whole, you know, as biologists, we're really interested in the community as a whole, and we take the mean of the community and the average, and but science is slowly starting to evolve, and we're starting to understand individuals and how individual behavior can be really important as well. And this is really great because it shows that there is a lot of variability among individuals.
Within the community, within a population, within species.
Exactly. So trying to take just the average of the population that you're studying.
Yeah. They also have feelings, you know?
They have feelings. Yeah.
Some of them are, not so nice, and other ones are really cool guys, you know?
And some are brave and some are shy, and there's quite a lot of research coming into that now. And also in fisheries and stuff, there's a lot of hypotheses that the fish you are catching are always the brave individuals because they are the ones that will go for the bait, whereas the shy individuals, they won't go for it, so you won't catch them.
Yeah, but that's true. And I remember when I was fishing somewhere in Australia or whatever, people told me, oh, well, if you go in that area, all the fish are shy. And it's not that there's no fish. At least that's what they said. It's not that there's no fish. There's lots of fish, but they're all shy.
Yeah.
Because the ones that are not shy are all taken out of the water, and so you end up selecting for the shy trait.
Yeah.
And then the next fish that will just be shy, and you end up having just a bunch of shy fish.
Yeah. Yeah.
And it becomes really difficult to fish them.
Yeah.
Which is a good thing for them. Yeah.
Yeah.
So you talk a lot about the fact that this is a nursery for stingrays and that you've monitored their movement. And so you also looked at their diet. Mm-hmm. So how do you combine the diet information from stomach content that you so beautifully analyzed in your kitchen, on your kitchen table, the stable isotope analysis, which are also used to determine where it is in the food chain, and then the telemetry, which is giving you information on its movement. How do you combine all that to make one PhD?
So that was the tricky part, but actually the data itself lended itself really well to turning it into a really complete story, because a lot of times you only study one aspect of the animal, you only study its diet, and you get a really limited understanding. But if you start to bring in all of these different aspects, it really gives you the big picture story. So I'll use one thing for example. So I was looking at juveniles of the 3 different species, and I found that they were eating different things, but then you bring in the telemetry data and you actually see that they are not spatially segregated, which means that they all live in the same area, they all co-occur together, they all live together, but then they're still eating different things. And so
that's really interesting information.
It's really interesting information. And then also adding in the stable isotope stuff, because that really gives you a longer term picture of the dietary trends of these animals through their sort of how they, as they age, as they become adults. And then we could see that the adults and the juveniles were eating completely different things. And then that again tied into the telemetry data, because the adults and the juveniles were living in different places. So it really just allowed you to understand what was going on a much bigger scale than if you were just looking at what they were eating.
It's fascinating. So you were able to kind of make more inferences, I guess, and say, well, it seems like this species of juvenile relies on this type of prey, and then this species of juvenile stingrays prefers this one.
But even though they all live in the same area,
so
there's something else that's driving that differentiation. It's not, now you can say we definitely know it's not spatial differentiation, there's something else going on.
So it's behavioral, or maybe they
morphological. Some if they have different dentition, one can crush harder prey, whereas the other one likes to eat soft worms, kind of thing. So
I like to eat soft worms. I mean, why not? Why not? I'm sure at some point human beings ate soft worms. We're talking about polychaetes, right?
Yes,
polychaetes. They are not the most appealing worms out there.
They're pretty gross, and they have I don't know if you found that as well, they have this really distinctive smell. They have this sickly sweet smell.
A sickly sweet smell.
It's not a great smell.
Well, to be fairly honest, the polycod have a stomach that is about this big. So the polychaetes that I found in there were very small. They don't eat polychaetes a lot. And when they do, they're really, really tiny. So the overwhelming smell of everything else that is in the stomach, half digested, takes over the potential sickly sweet smell of polychaetes.
Well, you have to come visit so you can.
I am absolutely
sweet aroma.
I am looking forward to sleep in your guest room with all these stomach content. Oh man, really, we'll have to talk about this. This is not gonna work.
I can remove them for you. We have,
Okay, good.
Actually, it has been my plan for a while to we have a storeroom downstairs.
Oh, good. Okay,
so
well, that will be a good
happen.
opportunity to move this thing out. So one more thing regarding their fieldwork. Because you spend a lot of time in the Seychelles, which is such a terrible destination, how much time were you spending there?
So it varied. So mostly it was sort of between 3 to 4 weeks at a time.
Okay.
I think my shortest trip was 3 weeks, and I think my longest trip was 6 weeks.
Yeah.
And I had 5 different trips in total. So
Oh, wow,
if
that's
you
a lot of
added it
trips.
all together, I think I spent 6 months there.
Yeah, okay.
Yeah.
And were you sleeping in the water?
No, so we were quite lucky. There's that research station on an island that's really close to the atoll.
Okay.
And that's where we would spend most of our nights. And that was where they had really nice accommodation and you had a bed and all that kind of stuff.
Yeah.
But then sometimes we would camp over in the atoll because...
Oh, you would?
Yeah.
Wait, where do you sleep?
So that was when it became much more rustic because it used to be an old coconut plantation. So there is sort of this building structure there, but it's really old and really run down.
Wait, so I'm trying to visualize. So this atoll is like a rim, right?
Yeah.
Like a volcano rim, but it's sand flats mostly.
And then there are a few small islands.
Okay, and they had coconut plantations on these islands?
Yeah, so just the biggest one. So actually the biggest island is called St. Joseph, which is why it's called St. Joseph's Atoll. And it's sort of almost like a moon-shaped island.
How big are we talking? Like, how big is it?
It would probably... it's not big. It's probably like 500 meters.
Okay, so it's really small.
It's really, really small.
Okay.
I mean, you can walk
and
through
somehow they
the whole
had a
thing.
coconut plantation there.
Yeah.
Okay.
Don't ask me why, but
That
they
is
did.
interesting.
Yeah. I mean, it was very small scale,
Yeah.
obviously. So there's sort of this really old rundown.
Oh, they have these giant coconuts there.
Yes.
Like...
Oh, the Coco de Mer, yeah.
Yeah.
So that's like their national plant, I don't know, but it's this ginormous nut.
So in French, they're called the butt coconuts.
Yeah,
because they look
yeah.
like a butt. They're so big.
Yeah. So, but no, these are just normal coconuts.
Okay, not butt coconuts.
But yeah, so they have this really old rundown building where they stuck a couple of beds in and you would sleep on the bed, but there's no running water, there's no electricity. It's really like camping out under the stars, which was really beautiful.
That must have been nice.
But there's no running water, so you there's like rain water collection tanks, and you take like a little bucket and you'd fill it up with water and you'd go have your little bucket shower on the beach around the corner where nobody could see you. So you're like looking out onto this beautiful expanse of turquoise blue waters and little green islands dotted everywhere. You're having a nice,
Wow. Is there a bioluminescent plankton?
No,
there?
so we never saw any of that. No.
Yeah. Well, can't have everything.
Can't have everything.
So this is absolutely fascinating. Everything's fascinating. It's a fascinating conversation. And you know what? You made me want to go to the Seychelles.
It's beautiful.
And to South Africa.
Yes.
In your room. In your bedroom with the stomach content. But I think we're gonna have to wrap it up because we could go on forever. In fact, we have gone on forever.
Yeah, I mean, I arrived here yesterday afternoon and we have been talking non-stop.
Yeah, basically. Yeah. We have not stopped talking for the last 24 hours almost. Pretty much. We did sleep a little bit.
Just a little bit in the middle.
But there's one more question I want to... there's tons of questions I want to ask you, but one for the purpose of this video, which is, you've got this great YouTube channel. And I'll put a link everywhere to this YouTube channel. Definitely in this description below. I don't like saying that. That sounds so silly, especially right now when someone else is listening to me saying that. In the description below, there shall be a link to your awesome YouTube channel, which stole my YouTube channel's name, but that's okay. How long have you been doing this YouTube channel thing for? Like 2 years?
I think it's 2 years. I think it's around 2 years.
Because I started following you about 2 years ago.
Yeah.
It must have been right at the start.
Yeah.
And at the same time, there was also Maria.
Yes.
Who was starting.
Yeah, we sort of started around the same time.
And I was just kind of thinking, hey girls, you're stealing my idea. I totally wanted to do this first. And then, I started a bit later than you girls. So how did you get into doing that? science communication
Actually,
thing.
it was funny in the beginning because I was just on YouTube and I wanted to follow the YouTube channel of another marine biologist. Like, I wanted to see what other people's experiences are like as a marine biologist, because it's such like a romantic field, and all of the notions and, you know, the public sphere is really different to what it's really like. And I just wanted to follow the journey of another marine biologist. But there was, apart from one other channel, which is also really good.
I think it's Tim, his name?
Yeah.
Or I'm not sure.
But other than that, there was nobody on YouTube as a marine biologist PhD student specifically. That was what I was doing at the time.
That has like a regular YouTube channel.
So I was like,
oh my god, I can be that person.
I might as well just go for it. And I've always liked sort of blogging and science communication, and I like doing presentations at schools for the children and just really spreading, you know, information about the ocean and about life as a marine biologist, because it's difficult to understand how the life of a marine biologist is.
And it's so diverse.
I
do such different things.
So just as a disclaimer, we're going to be filming another video after this, which is going to be on my channel, talking about the diversity of marine biology, career paths and that kind of stuff.
We'll basically be talking about how our degrees differ and our experience differ.
And our fieldwork and all that, because it's so different. So I just decided, you know what, fine then, I'll do it. I'll make a channel. And I was interested in science communication. It was a new experience. I'd never really done filming or film editing or anything like that. It was a new challenge. So I just decided to go for it.
Was it difficult at first?
It was difficult.
It's a bit weird to film.
It takes a while for you. And I still feel like I haven't quite gotten used to it. It takes a while for you to really get used to it.
It does.
So
and the worst I find is that once you start filming yourself and you get into it, it's fine. But then once you load that up and start editing, you're just thinking, oh my god. I don't want to have to stare at my face for hours to stare at me saying silly things
over and over and over again. And just the whole editing process and setting up your channel, the sort of you know, the impression you want to give off as the first thing people you see when they open up your channel homepage. What sort of impression do you want to give off and all that stuff. It's quite difficult to start off.
And that's the thing that I found particularly difficult, was to not make a fool of myself to the eyes of my peers and of professional professors and marine biologists and the professor who hired me for the PhD and research assistant position I have now. He looked at my videos before I even moved in, right? And I didn't know. Until,
it's like another form of social media that people can
until one of his students told me and said, oh yeah, he showed me these videos of you and you're so active and all that. And I thought, oh no, he did?
They've seen the videos.
And so that's what I found the most terrifying, is to put your image out there, and in a way you're kind of have to give up a lot of pride, because you're not, it's very hard to feel like the videos are accurately representing what you're thinking, or that you're just not being silly and looking stupid to the eyes of the community, or saying something stupid and someone else is watching the video and thinking, no.
It's much easier to picture the people watching your videos as strangers. But as soon as somebody you know comes up and says to you, oh, I saw your video, and you're like,
and I never advertised my videos on Facebook. And then my friends started doing it, and they tagged me in the video, and I was like,
don't do that. ...
That's right. I make those videos.
Untag. No, it is. It's a tricky thing, and it's something you do want to be proud of. You do want to share it, but at the same time, you do feel a little bit silly, and
it's hard. It's just hard to make. So it's hard to define the audience. It's hard to define to which extent you want to go into the science.
Exactly.
And if the audience is people who are, you know, looking into being marine biologists, then maybe you want to talk more about the exciting side, and you also want to talk about the drawbacks, but that could be something that is a bit boring to someone who's already in the field. Maybe someone who's already in the field wants more details about what you're doing, or about what you're feeling, or it's hard. What appeals most to people on YouTube videos is this kind of really personal human experience, and that's not what appeals to researchers. That's something that I'm putting it wrong. That's something that researchers try to hide.
Exactly.
They often try to hide that they're human beings.
Yes.
They are machines, and they go through this very thorough scientific process, and they don't have any feelings, which is absolutely untrue. And when you start putting your feelings out there,
as a scientist,
you
it's
become
terrifying.
vulnerable. Yeah. And it's a very tricky thing to do. Yeah. But we've both done it.
So are there if there's any other marine biologists out there who want to join our little community?
Yes, please do. And then maybe we can meet up and do these crazy videos. And, but it was absolutely fantastic to talk to you, Telly.
Yeah, thank
Chantal.
you. It was really, really great just being here and getting to know you and
I
meeting
am so
you.
happy we met, and it's really weird meeting someone that you've seen for 2 years on YouTube, and all of a sudden they're in front of you, and you're just like, oh my God, what do I say?
Real person.
It's kind of like meeting someone famous.
Yeah, well, I mean, I'm definitely nowhere near famous, but
it feels like that.
Yeah,
like it feels like this person exists on paper or on the screen, but they don't exist in reality, right?
Yeah.
So no, it was awesome meeting
thank you very much.
And I look forward to visiting you in South Africa.
Yes,
yeah,
it's gonna happen.
Yeah. So thank you guys for watching this.
Yeah,
and go check the video on Telly's channel.
Yeah, we'll also do one. It will be a bit of a shorter one, covering a little bit of a different topic. So.
Well, we're saying it'll be shorter, but
it's
probably going to be another 2 hours.
I have a bus to catch in 2 hours, so it has to be shorter.
Yeah, I've got lots of lab work to do.
But yeah, thank you so much for having me.
Thank you. Yeah. All right, bye.
Bye.